So we are continuing our discussion a little bit on methylation and epigenetics. Remember, we talked about me and you having a genome, which is all of the DNA, but your epigenome is not only all of your DNA, but it's the specific patterning of inhibition that could exist at another layer on top of your actual DNA to determine if genes are turned on or turned off. And some of these things can be controlled by the environment, and actually be passed on to your offspring. So this is new stuff that has kind of gone against the idea of you only are whatever you've inherited in your genes. And if anything you do in your lifetime that's stupid won't be passed on to your kids. Now we're finding you could be doing stupid stuff that could be passed on, and these particular changes in your gene expression can be solidified biochemically into your DNA and actually be passed on in sperm cells and egg cells, and therefore affect your kids or affect future generations of your particular family. My Lee family could be forever and ever tainted by the things that I've done during my life. Anyways, still a lot of evidence. We're trying to figure things out. So we talked about in the last video that one way that a kind of biochemical marker that can be added is called the methyl group. And a methyl group can be added especially to the cytosine bases. You got A's, C's, T's, and G's. So on cytosine bases, there can be methyl caps that show up there. And usually these methylation caps will actually prevent that particular gene from being expressed. So it's inhibiting transcription or switching off that particular gene. And eukaryotes have lots of methylation going on. Even when you're studying regular human beings, you can see that the relative methylation level, so how many of these caps actually exist, greatly changes as we get older. So it's almost like when you're a kid, okay, we don't need to turn on all of those genes, and then when you get to 26 years old, or as you start going through puberty, hopefully you're not going through puberty at 26, but that's somewhere in between, that some of these genes will be turned on when they need to be turned on, and so that this kind of methylation is going to disappear. So in the last video, what we did talk about is that this is a normal process that's supposed to happen, but the key point we're trying to point out here is that whatever you do in your life, the environment can actually affect your methylation patterns, and those methylation patterns could be passed on to next generations. So similar cells in a tissue are expected to have the same methylation pattern so that they can act like same types of cells. It's kind of like differentiation and specialization. All my skin cells next to each other are expressing the same genes so that they can all be skin cells and do what a skin cell is supposed to actually be able to do. So one last time, just to rub it all in, environmental factors can influence the pattern of methylation and gene expression. And it's probably one of the reasons why, and I mentioned this in the previous video as well too, when you have identical twins that grow up, they have the same genome, but they may not have the same epigenome. What they expose themselves to during their lifetime, especially if they grow up apart or grow up doing very different activities or different jobs or different types of sports, you can end up with differences that result through that particular process. So we call it the epigenome. So identical twins out there, if you want to be truly and identically the same forever and ever, you guys should make sure that you do the exact same hobbies. You know, every time you do something bad, the other person does something bad. I don't know what those bad things are. I don't do those things. But just in case you do anything bad, make sure your identical twin does the exact same thing so you can stay identical forever and ever. I wish I had an identical twin.